We are pathogens, and we've just invaded the fluid of the human body.
We want to get past the immune system and infect as many cells as possible.
I carry an antigen, which is a unique protein that causes a specific immune response in the body.
This antigen is unique like an ID card and can be recognized as foreign by other cells.
I am a macrophage, a phagocytic cell that ingests pathogens. I am the on-site security guard who looks out for invaders, and I inform the higher level officers. I noticed the pathogen's ID card or antigen was non-self, so I digested it through the process of phagocytosis. Hey, let me see your ID card.
Never.
No.
Hey, this is non-self.
I'm a class 2 MHC molecule, and I bind to the foreign peptide, or the antigen, and I transport the antigen to the surface of the macrophage, so that other cells can notice the non-self antigen. I'm a very important aspect of cell-to-cell communication in the immune system. Here, officer, is an invader's antigen. It does not belong.
Do you really? Let me see what you have. Here, grab onto this rope. I am a helper T cell, and I become mature in the thymus. My job is to help alert certain molecules in the immune system that there is a problem. CD4, or the rope I have, enhances the binding between myself and the MHC molecule, which contains the antigen on the macrophage. Once I receive an antigen, I proliferate and secrete these markers. Okay, or interleukin-2, which is a chemical that acts as a signal to activate B lymphocytes. Hey, wake up! I noticed you have ID holders. We need you to help find more of these ID cards.
I am a B lymphocyte, and I have been activated. So I proliferate by calling in other FBI agents. Then I go to transform into a plasma B cell or a memory B cell. I am a plasma B cell now, and I am secreting antibodies. These antibodies act as handcuffs by attaching the antigen and keeping it in place for other cells to kill.
I am a neutrophil. I'm also known as a white blood cell or a leukocyte. That's Leukocyte. I have been attracted here by the antibodies. I kill the pathogens by attacking and digesting them through phagocytosis.
And I am a memory B cell. I'm able to remember the antigen of the specific pathogen, so I make a big stock-like of the antigen. Next time the pathogens invade, the immune response will be quick and the antigen will be already made.
Fiend? End? All right. All right, so now it's Q&A time, so don't go anywhere. Audience, take a look at the actual sequence of events. Think about what you saw. Are there any questions about the roles that people played and how the process unfolded?
That's, the CD4 helps enhance the binding with the MHC molecule, which has the antigen on the macrophage, so it just helps them bind together.
Okay, so it's the antigen to the macrophage?
No, the rope is a CD4 molecule. It just helps bind the helper T cell and the
MHC on the macrophage with the antigen.
Yeah.
Now, what is a CD of any type? CD4, CD8. What is that? It's a molecule, but... No, I don't need to know what it stands for. Just what is it?
It's just like a molecule on the thing that like connects cells together, I think.
Okay, so CD is a receptor. It's a receptor molecule. Okay? So if you hear CD4 or CD8, that's a receptor. What do CDs bind with? What do CDs bind with? What was that?
The MHC molecule.
CDs bind with MHCs. Where are MHCs located?
On the surface of the macrophage.
Or on the surface of normal human body cells. And then where are the CDs located?
Within the cell.
All around the bloodstream.
Just floating in blood? So this is kind of important to know where these things are. So where are they, audience?
What was the question?
Where are CDs and where are MHCs?
Well, MHCs on the surface of the cell, and the CD4s are
Yeah, CDs.
from within the cell.
CDs are receptors on T cells. Mainly.
Helper T cells,
Helper
right?
T cells, right?
Right.
Okay.
Right, but you could also have CD8s and CD4s. CD4s are on helper T cells. So CDs are the receptors on the Ts, and they bind with MHCs. The MHCs, whether they're on macrophages or whether they're on normal infected body cells.
And then the T killer cells of the CD8.
Right.
T killer cells.
But there's no key killer.
Yeah.
That's the next pathway, though. You're right, once we get there. Now, it's not critical that you understand what CD8 is versus CD4, what MHC1 is versus MHC2. For this test, we've made it a little bit simpler. We just want you to know what an MHC is, and we want you to know what a CD is, and how MHC and CD associate with one another. Okay? Now, if you want to know the details, it's actually pretty easy to remember what goes with what. CD4 goes with class MHC2, and CD8 goes with class 1. So if you take those numbers and you multiply them together, you should always get 8. 2 times 4 is 8. 1 times 8 is 8. That's not the reason why they're named that. That's just a way to figure out what goes with what, an easy way to remember that. Okay, so there's a lot going on here. What don't you understand?
Please report to room 2400. Allie, turn to room 2400.
Okay, yes.
Why is it called a plasma B cell after it proliferates?
Oh, it differentiates into a plasma B cell.
So like transforms.
It becomes more specialized in
Okay.
it either turns into a plasma B cell or a memory B cell. Plasma secretes the antibodies for the first part.
The specific antigen?
Yeah, and the memory B cell will remember it so that next time this whole process doesn't have to happen, but it already has it, so it can attack right when it sees
Secondary,
it.
and it's a lot quicker.
Okay.
The secondary immune response.
So B cells obviously don't have brains. What do we mean by memory?
They make a lot of the antigen that attack the cell. So,
Antibody.
a lot of the antibodies for the antigen that attack the cell. So the next time the pathogen invades, it can more quickly wipe it out. So
Now, where are the antibodies in memory cells?
It's on the surface.
Yeah, it's on the surface, so it's displayed so that all of them.
It's embedded in the membrane. So is there just one memory cell after an attack?
No, there's literally millions.
So why is the secondary immune response, the second time the body sees an infection, why is it so much faster? Because there's memory cells.
Because there are already antibodies like built
Made
up
for it.
For that specific,
beforehand. For that specific pathogen/antigen.
Okay, so what happens if there's no memory?
And then
you have to go
do the
through the
process
process
again.
all over again.
And then what was that sequence of the process, just summarized again?
So the macrophage comes, and then the MHC binds with the helper.
First, the macrophage engulfs the
The
pathogen.
pathogen, yeah.
And then the MHC displays the ID code.
Yeah,
Or
the
like the
antigen
antigen.
of the pathogen.
Yeah, and then
then
the helper T activates the B lymphocytes, which can either
Make
become
antibodies.
a B plasma making antibodies, or it can become a B memory cell, which displays the antibody at the surface, so that next time when it comes across the same pathogen, it doesn't have to go through
through the process again.
And then the antibody locks the antigen for the neutrophil to come and destroy the antigen from the pathogen.
So if there's a memory cell, you can avoid all that, and it goes right from memory cell to what we call monoclonal expansion. The memory cells just start cloning and making tons and tons of plasma cells, pumping out millions and millions of antibodies. So the second time that same pathogen comes in, you might not even get sick. So secondary response is much quicker. Okay, I heard some talking out here, so I know that there's some questions. Aiden, did you guys have some questions? David?
We were just wondering how the antibodies kill the antigen.
They don't kill the
Right.
It's kind of, we use the example of like it's kind of handcuffs it
Right.
for the neutrophil to come in.
That's what we were
destroy it.
discussing.
It
Now-
basically stimulates the response
From the neutrophil.
Now there's a problem with using the word antigen in place of pathogen. So every cell has its own antigen. All of your cells have antigens. Like your blood cells have A antigens and B antigens. We call those self-antigens, something your immune system recognizes. Pathogens, the bad guys, they have antigens, but your immune system doesn't recognize those. They're the wrong ID tags. So when we say antibodies kill antigens, that's not necessarily right. An antigen is just a protein. You can't kill a protein. But you can say that antibodies kill pathogens, which would be like bacteria. So antibodies bind with antigens, and in that process, that can lead to the killing of the pathogen. So they can just stick to them and activate something called the complement pathway that pokes holes in the pathogen. They can just stick to them and keep them from reproducing, or they could stick to them and clump them up in the lymph nodes, where it's easier for your immune system to get at them in a group. Hopefully that makes sense, that it's easier to go ahead and kill these things if they're all bundled up in one area than if they're just all floating in disparate places in the body. Yeah, Laura.
What different pathogens does this work for? All of them, or is it like just viruses or bacteria?
This is a specific immune response, so it's basically different for every specific type of pathogen that enters the body.
But the process is the same for them all.
Yeah, the general process is the same.
Except for HIV, which messes up the helper T cells and then nothing can work.
Right. So in our next group, they're going to talk about cell-mediated, and you'll notice that when they talk about their pathway, they're also talking about helper Ts. So I want you to really understand the connection between these two pathways. The helper T is the one that coordinates both. So they both typically happen at the same time concurrently. It's not that one happens before the other or one leads to the next. The general makes both pathways happen. All right, any other questions? Oh, yeah.
Can you explain what IL-2 is?
Interleukin-2 is a chemical that is secreted by the helper T cells that serves as a marker, a signal for the B lymphocytes to proliferate.
So like the marker that was thrown.
Right. So in this skit, we use the markers. It's just something. It's just a signal. It's an intercellular. It's a cellular signal.
It's a specific type of chemical signal. The generic name for chemical signal is chemokine, or lymphokine, or cytokine. Anything with a kine just means chemical signal. And so interleukin is a specific type of one of those. Interferon is a specific type of those. So you can lump all those words together: cytokine, chemokine, lymphokine. They're all general terms meaning chemical signals. And then you have specific examples that we'll talk about, like interleukin, interferon, stuff like that. You won't need to know the difference between interleukin 1 and interleukin 2, but just know in general what an interleukin does. And you can. The nice thing is you can kind of break down the word interleukin.
Inter between leukin and leukocyte.
Yeah, you got it. Any other questions for this group? All right, thank you.